All right, I'd like to welcome you to our last session. So thanks everyone for sticking around for these 2 days, and we really appreciate everyone's presentations and the opportunity to interact with all of you. So I'm Kathy Ferreira. I'm going to be chairing this next session, and I'm going to be your first speaker as well. Oh, and so we will have a total of 4 speakers. Bosu Huang will be talking to us about protein design. Craig Levin is going to be talking to us about novel technologies. And Florence Jensen is also going to be talking to us about the latest in technologies for imaging. We'll follow that then by closing remarks, and those remarks are going to come from Helen Nadel and also from Andre. All right, so I'm going to be telling you a little bit about something that I'm really passionate about, which is being able to choose targets and design ligands for novel radiopharmaceuticals. First, you've probably heard that that we're all the molecular imaging program at Stanford. So who are we? So I am the division chief. I took over from Sam when he passed away. And we are a group of now 41 faculty members who span many different departments at Stanford. So everything from chemistry, radiology, radiation oncology, engineering, a very broad group of people, and on the order of about 300 trainees at this moment as well. So this was really Sam's vision of a group that would come together to develop new ways of imaging inside living systems. And we're continuing really to thrive in many ways with new facilities. We have educational programs, workshops. So anyone who's not part of MIPS and would like to become part of MIPS, please let us know, and we're a very welcoming group. You can also see that our group includes many of our colleagues from nuclear medicine as well, and so it's really been a great opportunity to bring people together. So I wanted to touch one more time on opportunities to interact with Sam. So one other thing I hadn't really heard people mention as much, when my dad was ill, I was still at UC Davis, faculty member at UC Davis, and I reached out to Sam, and he helped me to arrange for surgery for my father, but surgery wasn't successful, and I reached out again to Sam, and within hours, he was able to connect us with an oncologist who could help things to move forward. So he was really, this was my email, it was remarkable, his ability to make things happen and to really work with people and care about people. I was then a visitor in his lab, and then moved to Stanford in 2018 and became division chief. You've seen this slide before, so as a program, we're really thinking about what do we want to be, and how do we follow on with Sam's vision, but also move things forward in new directions. And so one of our real goals is to be able to bring together target discovery, ligand discovery, first in human studies, new technologies, et cetera, into a single program. I'm going to be particularly focused on the development of methods to choose targets and ligands in this talk. So part of what our laboratory has been doing and moving forward is to combine spatial transcriptomics and spatial proteomics to be able to better understand biology and better move the field of molecular imaging forward. And the idea is that, we've done bulk sequencing for many years in the lab, and there's a lot of information that you can get from just pure omics. But you realize that as many of us, or most of us, are probably imagers, right? And as an imager that there's a lot more information that you can get even from a single image. So this is an image that came from my next door neighborhood of where you could have, with omics, if you had been able to go into that spot, you might have been able to tell that there had been wheat there and there was evidence of a coyote. But you would not know that that coyote had jumped onto someone's car, stolen their loaf of bread, and run down the trail with it. But this image gives you a sense. So people who wondered where did their bread go, they had an image actually, and it really told them what happened. So there's a lot more information here that we think that we can gather, and it really inspires us to develop new technologies to try to move that forward. Just another example where omics might tell you that there had been squirrels there, but you look at this image and you really think there's something very interesting going on, and you'd really like to know more about how those squirrels are interacting with one another, and what the scenario was for them. So just to give a sense that we all believe that in the power of imaging and the power of imaging to do new things. So as we apply this to omics, we have a number of questions that we would like to ask. We think that spatial omics can improve our ability to choose targets. It can, we in particular in our lab have been focused on finding invasive cancer cells in pancreatic ductal adenocarcinoma. We are also now looking in prostate cancer to try to better understand the set of markers that are associated with high Gleason scores. And we want to really understand the spatial correlation of different cancer markers. So in order to do this, our pancreatic data set looks something like this. So we've collaborated with folks in various departments who have surgical samples that they're able to share with us. So IPMN is a precancer for pancreatic cancer, so we've taken surgical sections. We now can take FFPE, so paraffin-embedded sections, and sequence these. With Visium, which is not a perfect technology, and still this technology of spatial sequencing is still developing, but you're able to, the really good thing is you can look at a depth of 18,000 genes in each spatial location. You don't have single cell resolution. You have between 1 and 10 cells within every pore, but you can learn a lot about the spatial distribution. So we've had a total of about 40 patients whose data that we've looked at from the spatial omics platforms, and then we also have augmented that with the large data sets that give us another few hundred patients with pancreatic cancer to better characterize molecules and targets that we might be interested in imaging in the future. And what we've learned, if we just take an example of this pancreatic ductal adenocarcinoma, it's really a reminder for us that this is really a ductal disease, that the cancer cells are traveling through the ducts. And our favorite marker at the moment that we're pursuing for molecular imaging is one of the tight junction proteins, Claudin-4. Now, if you look at Claudin-4 expression, you can observe that these ducts are really filled with cancer cells that are moving with very high density, that are moving through the ducts to spread disease. We can augment this with immunohistochemistry, where we can visualize various markers. But you can see that this is mRNA expression of Claudin-4, for example, on the outline of that duct, and you can observe that the expression is significantly increased in the regions that contain these ducts that are really filled with cancer cells. Now, if we compare that to other markers, so there are some other markers that look encouraging. We use S100P as a gold standard here. S100P is mostly an intracellular and somewhat secreted protein that is only expressed in cancer in the pancreas, so it's somewhat of a gold standard. MUC5AC is expressed very early, and TFF1, similarly trefoil factor 1, come up relatively early in disease. Claudin-4, we found, comes up at the time of invasive disease. And we've looked at this compared with, for example, alpha V beta 6 or beta 6 integrins and FAP, fibroblast activation protein. You can see there is signal there in each case, but there isn't signal that on a gross scale is associated with these ducts that contain cancer cells. We can look at other questions too, like insulin expression. So we can, for example, also look at how the pancreas is functioning by comparison through these studies. So this is the kind of thing that we're really wanting to be able to do. We also can then take high-resolution immunohistochemistry, and we can compare our markers to the way that pathologists would traditionally look at these sections. And we can recognize the cellular morphologies that are associated with invasive disease. So we can spatially correlate all this information, and we can really make sure that the markers that we're associating with invasive disease are those that pathologists would agree correspond to those spatial regions. So the marker that we've become, one of the markers at least, that we've become most interested in is Claudin-4, which is a tight junction protein. So one of the other good opportunities about some of these other alternative markers are that they are not necessarily accessible in normal tissue. So Claudin-4 is a tight junction protein. It's really expressed between and holding together epithelial cells in normal tissue. It's only when disease is present, and particularly cancer, that the tight junctions open up, expression is increased, and the protein also becomes accessible. So we do find that when we've made an imaging agent and we go to image it, we don't see the accumulation of our molecular imaging marker in the regions that have normal tissue. So expression is upregulated about 16-fold, and it depends. So we've tried to quantify the fraction of what we would call cancer cells that do express this marker. Depending on what we call a precancer and what we call a cancer, it's anywhere between about 90 and 97% of the cancer cells on that journey from precancer to cancer that express Claudin-4. And we have gone back and looked at the phenotype of those that don't express Claudin-4, and we've shown that those are the cells that don't have an invasive phenotype on immunohistochemistry. We also have looked at the area under the curve. I'll show that in just a second. But so how this works, the kinds of things that we do, we create heat maps that allow us to look at gene expression. We particularly focus on cancer cell surface markers. So we're looking for molecular imaging agents that we can develop based on the expression of things on the cell surface. James Wang, who's been taking all your pictures, has then created a cancer surfaceome score, and using that score, we're able to score different markers in terms of whether they are spatially correlated with cancer and are a cell surface marker. One other thing that is really interesting is we can look at the spatial correlation then of cells that express these cancer-associated proteins. And we find that the more invasive or the higher grade the lesion, the more highly correlated you have a group of proteins expressed. And so in each cancer, we can go in then and look at what is this group of markers that are tightly correlated, which of these would we like to image? It gives us a number of different alternatives, or we can think about imaging more than one of them because they do change their expression level at different times. So it gives us the opportunity to ask those sorts of questions, as well as looking at, so the p-values in these studies are like 10 to the minus 300 often, really super high. We were really, I think, very surprised to find that there would be one marker in a disease like this that would be upregulated so consistently across patients. Yeah, so what fraction? Up to 97%, depending on exactly where you draw the line. The area under the curve for a diagnosis of PDAC compared to normal for this as a marker is, again, is 0.9, and better than other markers that we've studied. And we also have looked at pseudotime. So pseudotime gives us the ability to map out the progression from a precancer to a cancer. And what we found with Claudin-4, it's kind of hard to see on the IPMN, which is the precancer, but there's a straight line there that's the red, bunch of red pixels there on the end of the graph. And that's basically telling you that there is a very sharp curve where Claudin-4 becomes upregulated in the transition from precancer to cancer. So this is still So pseudotime gives you a way of visualizing that journey that cells go on and to really think about where your individual markers stand in that transition from precancer to cancer. We also can think about using spatial expression. We also can think about how effective a radiopharmaceutical, a therapeutic radiopharmaceutical might be. So we can take the mRNA expression of the target. We can take what we know about the relationship between RNA expression and protein expression, which for cell surface markers tends to be pretty high. And then we can map out the spatial distribution that if we knew the binding affinity or the fraction of cells that would take up our target, we can then do that mapping and we can predict what the radiation dose would be. As I said earlier, this isn't a perfect technology, though. There's a lot we can learn from single-cell analysis going forward to looking at protein and looking at individual cells. So CODEX gives us the ability to do that, and we've been augmenting our Visium studies with CODEX, where we can look at individual regions on a single-cell level, and we've learned a number of different things. So the yellow circles that you see in region 4, the lower left area, that yellow surrounding is potoplanin, which is similar to FAP. It's a marker of activated fibroblasts. So there are activated fibroblasts there, but they are in a thin line surrounding this duct that contains a large number of cancer cells. So if you're able to get down to single cell resolution, you do see that there are activated fibroblasts there, but the distribution there aren't as many cells, and the distribution is really different than perhaps you could achieve if you were to directly target the epithelial cancer cells themselves. So that's why we've taken this path of directly trying to target the cancer cells. We also have found that there's a lot that we can learn from the spatial biology about immune cells. So we originally thought we wanted to come up with either a stromal or immune cell mapping method and strategy. And what we found was that there are tertiary lymphoid structures that are very frequently present within these tumors. That's what you're seeing here. It looks like a lymph node, but it's really a response of the stroma to the invading cancer. And so in many of the patients that we've looked at, there's an overwhelming number of immune cells that are related to these tertiary lymphoid structures. And so we need to be very careful in our ability to interpret the immune cell imaging data that we acquire. So our next talk is going to be from Posu Huang. Posu and I have been collaborating then on taking these targets and trying to generate peptides from them. He's going to tell you just in general about his work, but we have ongoing work to be able to identify and optimize particularly cyclic peptides against these targets. And I don't know that I have to convince this audience, but peptides really have this is actually a slide that I got on the left from Sam. Peptides really have a great opportunity. If you have the same binding affinity, if you look as a function of molecular weight, the smaller ligands, the very small peptides, have an even better opportunity to bind and to adhere to your target than an antibody because of the fact that they can extravasate, they can find their target so efficiently that they're really not blocked by other cells. So we really believe in this opportunity. So we have then gone ahead and created, Jay So in particular has really taken the lead on creating a radiopharmaceutical for pancreatic cancer based on Claudin-4. We've worked with Laura Attardi here at Stanford, who has really nice mouse models of a adult onset pancreatic cancer, and we've demonstrated that we can image and detect even very small metastases within both in the pancreas and metastases that are moving through the peritoneum. This was, really, I think, a good surprise for us. We're able to get on the order of 20 or 25% injected dose per cubic centimeter, so really a good dose into this mouse model. Then just because this is such an audience that's so interested in prostate cancer as well, I wanted to just say something about high Gleason score imaging. And so we've had the opportunity to go back and take some of Andre and Hung's samples from patients that have been imaged with PSMA or with GRPR and do spatial transcriptomics on these and get high resolution immunohistochemistry as well. And this is just an example of a case where a patient with a very high Gleason score, if you look at the immunohistochemistry in the lower area, you can see it's the high Gleason score cells are circled. They have that invasive looking morphology associated with them. And we can then look at the signals that are present, the mRNA that's enhanced on those particular spots or those particular cells that have the high Gleason score. So in the upper left, that's the gene that for PSMA, FLLH1 is the gene that encodes PSMA. You can see that it is upregulated, but it's not specifically and very sensitively enhanced on the high Gleason score cells. And in fact, there are other markers. The top right is an example of a marker that is very highly enhanced specifically on the high Gleason score cells. And again, we've looked at the spatial correlation of the markers that have that kind of a signature. So in conclusion, I hope I've convinced you that there's really an interesting opportunity for spatial sequencing to provide spatial context for understanding gene expression. We think that transcriptomics can define a signature associated with different cellular morphologies. We've then selected markers and created peptide ligands, and we've been able to show that we can then develop a radiopharmaceutical with high accumulation in tumor and metastases. And our next steps are to create a theranostic agent and to hopefully translate this work in the near future. So I'm just going to give a very general thank you. Many of the people in my laboratory and many collaborators around Stanford have contributed, and for some reason that slide doesn't want to play. But yeah, but I really am indebted to Sam for giving me this opportunity, and really for many collaborators who work with us as well. And thank you so much for your attention. So Posu, do you want to get in the process of switching computers? And I can maybe take a question while we're doing this. Yes?
Correlate with local aggressiveness and resectability of the tumor?
So it definitely, on a small scale level, it definitely correlates with the invasive signature of the cancer. Yeah, and the only problem with pancreatic cancer is that it's uniformly, when it's found at the stages that, for the most part, that we've been looking at, lesions do become invasive and over time. And in the precancers, it does correlate with the other markers that we have for disease progression. And also in the prostate cancer, we definitely see that the high Gleason scores have and the markers that we're looking at on the high Gleason score cells are associated with very fast advancing cancer.
Is it an independent prognostic factor for outcome? Or does it just correlate with grade, and grade would be just as good?
It's a much more, I guess the thing is, we're finding that these are much more sensitive because, most of our analyses historically have been on a millimeter or multi-millimeter scale. And here we're really going in and looking at the cellular phenotype down on a very small scale, 100 micron spots. And we can differentiate, we can see whether there are regions on a 100 micron scale that have that invasive phenotype. So it definitely has the opportunity to provide information, I think, as we design new radiopharmaceuticals that are different, that is different and complementary to what we've had.
It'd be nice to compare early stage resected cancers to unresectable tumors to see if there's a different distribution.
Sure. Yeah, yeah. Definitely, we are able to look at precancers and what we know of as invasive cancers and demonstrate. Yeah. So thank you so much. And Posu, are you going to come up here? So just to introduce, Posu Huang is an assistant professor in bioengineering here at Stanford, and he's going to be telling us about novel molecular and generative design. So thank you.
Yeah, thank you very much for the introduction and invitation. So I unfortunately have felt too ambitious in covering the topics that I would like to talk about, but and I have a lot of slides, so maybe I'll just jump right into it. So sorry, clicker. So yeah, if we feel ambitious, I'll cover the second topic, which is related to the movie that Kathy showed in her slides about designing these cyclopeptides. But mainly, I'll focus on developing this platform called tracers. And unfortunately, this is also the audience where this acronym is a little bit confusing because there's a lot of tracers. But it is actually a platform that we now develop to try to bind to a peptide-carrying MAC specifically. So the question really comes to this notion where if you want to develop a targeted immunotherapy, the biggest limitation is actually to develop new biomarkers. And how do you actually access all these biomarkers? And this picture essentially just illustrates a few of the current clinical trials back in 2018, where people are essentially using antibodies and try to discover biomarkers on the cellular surface. And this really has significantly revolutionized the strategy. But the limitation of this strategy is that you essentially are missing out on a lot of the disease-coding antigens, which are potentially from this intracellular space. So my lab has been focusing on developing this technology to see if we can actually open up this possibility to access all these intracellular targets. So in this simple cartoon, this is just to represent the MAC1, how it incorporates these peptides onto the surface of the molecule. So you can kind of see that essentially every single cellularly expressed proteins will get degraded. And as they are being degraded, a fragment of it will get imported into the ER and tested on these MAC molecules. So on the lower right-hand side corner is more of the conventional approach, right? You have these surface proteins, and you have antibodies that you can develop and then use different modalities to actually interact with this marker that's on the cell surface. But MAC represented this unique opportunity, because now you can potentially access these aberrant proteins. Like it could be an oncogene product, and it could also potentially be from a viral infection. And we want to see if we can actually come up with a general solution to interact with these molecules. So, and of course, traditionally, this type of interaction is controlled by the T cell receptors. And but in terms of using this type of modality to interact with the cells, of course, now with this picture, you can kind of see that if you have this little arrowhead-like molecule, you can potentially use it for different modality. And as I mentioned, that this is critically in biology, right? Because MAC is essentially the central information hub to activate the immune response. And through this process, it is actually in the center of controlling all these disease responses, like including cancer, viral infection, and autoimmune diseases. And of course, there's involved different types of MAC molecules, which I'll cover in a second. So traditionally, as I mentioned, that this type of interaction has been the focal point of TCR engineering. So T cells receptors are the natural receptors for the MAC molecules, and they bind in this structural fashion where you have these CDR loops that actually land on the MAC molecule, and then there will be sequences that are essentially compatible and then can actually respond to the antigen that's presented on the surface. But as I mentioned, that there are different types of MAC molecules that are reporting constantly the physiological conditions of the cells. So there's the class 1 molecule, and essentially you can see that in the picture there's this cyan peptide that is a little bit shorter and is actually both ends into the hosting protein of the MHC molecule. And there's the class 2, which is normally expressed on the antigen-presenting cells. And it's historically been associated more with the autoimmune diseases. But in recent days, I think the prevalence of class 2 molecules has been increasingly getting a lot more attention because they now are responsible for a lot of the new discoveries that we have now found interesting. For example, microbiome interaction or the brain, gut-brain axis of pathogen transport. And also MHC2 are also now found to be highly expressed in some tumor cells. So, and of course, there's also this non-classical MHC called MR1. And essentially, instead of presenting peptides, it literally just grabs a small chunk of cellular metabolite and loads onto these type of molecules, so you can also monitor the metabolic states of a cell. So we set out to develop platforms in order to actually essentially replace TCR, because we think TCRs are a little bit too difficult to engineer. But with these three different classes, I'm only going to focus on discussion of the class 2 today, but we actually have a platform for each one of those. And this just to give you a picture of why this MHC class 2 is somewhat difficult to deal with if you're using a TCR, right? So in the middle of the picture, you can see that there's these CDR loops, and there are 6 of them. So if you are in the molecular engineering area, you normally would make mutations to these loops. So if you have a specific target, and if you want to build a binder de novo, it's incredibly difficult because you have to deal with 6 loops with a lot of residue, amino acid residue to mutate and then try to sample that space. So normally, the TCR engineering area started out with harvesting a known TCR that's responding to an antigen, right? So, but unfortunately for MHC class 2, this process is even more difficult because normally the class 2 peptides bind it's weaker. The TCR binding to MHC class 2 are binding weaker than their MHC class 1 counterparts. And the secondly, normally, at least again, historically, MHC class 2 is associated with autoimmune diseases. And those autoimmune peptides essentially just cross the binding threshold ever so slightly. And the TCR that's actually causing this autoimmune disease binds to those antigen peptide at an even weaker level. So this is actually really difficult engineering problem to solve if you have a given peptide and you want to just create a binder to it with high affinity. Okay. So we really want to solve this problem, and we went back to the drawing board and then actually literally list all the properties that you think if we completely ignore how nature solves this problem, because TCR is great, like in the natural immune response, because we have the thymic screening process, so you can actually remove non-specific interactions and so on. But if we actually just want to turn that into a theranostic agent, that's probably the wrong platform to go with. So we went back to the drawing board and list all the specifications, and the ones in red are like the ones that we think are probably of utmost importance. And mainly, we come up with this very simple design. Of course, it's just a cartoon, not yet the design. But we think that the binder would definitely have to have 2 components, right? One is that this binder has to be very specific to MHC class 2. So you cannot just have a platform that just goes onto your cell surface and then stick to every single protein on the surface. So it has to have this shape complementarity just to that scaffolding of the MHC. And of course, goes without saying that in order to accommodate all the different peptides, the ideal scenario is that if you can actually simplify, instead of having to use 6 loops to bind to some antigen of interest, you can just have one contiguous segment. That would make engineering life much, much easier. So, and of course, you can, if it's a protein, you have the termini, you can derivatize in any kind of modality that you want to deliver it. And we come up with a solution. And this almost came as a serendipitous discovery. So we happened to be developing some other platform in our lab, and we came across this molecule in gray here. It's a superantigen. So by definition, the traditional sense of a superantigen is a viral or bacterial protein that essentially cross-links MHC2 with the TCR. So it's a molecular glue. So the reason why there are superantigens is that if you actually form this molecular glue junction, you can actually activate T cells non-specifically. So it can actually bypass the antigen specificity for TCR and activate T cells, especially CD4 T cells. And it turns out in all of the structures in the PDB, the protein structure database, there is just one of this example where you have this protein literally sit right in between these two molecules. There are many other superantigens, but they don't bind the same way. So we're able to actually just take this one protein and we cut out the parts that are immunogenic, and we are left with this very simple, elegant design where you just have a helical bundle sits right on top of the binding groove. And we essentially treat this platform as if it's just a simple two-component system, right? You have a helical bundle scaffold that is very easy to make, and we have a lot of experience in engineering even from scratch. And we have this other element, which is actually very unnatural and very unusual. I call it the antigen recognition element, but essentially it's just a malleable loop on the surface of this helical bundle. And initially our motivation was just to see, can we actually just turn this loop into an antigen recognition component? Because the natural superantigen is agnostic to the antigen peptide identity. So this is a very brief description of how we experimentally test this. So we used a system called e-surface display. So you can see on the upper left-hand corner. So our protein will be the sort of like a yellow blob. It will be displayed on the surface of yeast. And then we use the MHC, tetrameric MHC molecule to detect for binding. So you're going to see some data points that looks like the ones on the right-hand side. So you can see that when you have the binding versus non-binding signals, you will have this double positive population when you have a binding signal. So, and we essentially just goes to this molecule. And then of course, there's actually a lot of engineering that has to go into this. But we ended up with this very simple cartoon representation of the story, is that on the lower left-hand corner, right, you have this helical bundle with a loop stretched across the surface. And as a first proof of principle, we literally only take 5 amino acid positions and randomize it. So you can imagine this is literally just a combination of 20 to the power 5. It's very, very small in terms of library selection schemes. So we can just use this and then put them on the yeast and use these tetramers to screen for binding, just to see if this system works. So, and it turns out we could actually get, for MHC2 molecules, you can get different types of targets directly from the NIH Tetramer Core facility. And so now in this plot, you're going to have, after we run through the screening process, we will isolate here 4 different binders. So the columns are the rows are each individual sequences that we have, and then the columns are actually the different targets. And once we build this system, you can actually see that we can get bindings are very, very specific on target. So again, this is actually was just first try. Like when we did it the first try, we can already get very specific binders. And of course, there are some low levels of cross-reactivity, and we can actually clean that up afterwards. So, and you can see that we have, we're able to actually get very nice binding curves. So these molecules are actually very stable and expressible. And we have binding affinity from low nanomolar or mid nanomolar range to all the way to the micromolar range. In this case, it's all nanomolar. But there are some targets that are more difficult. It's actually in the micromolar range. So, and we solved the structure. Because initially, we were thinking, oh, the scheme is just too good to be true, right? Because you now completely has a platform where you can just engage the site very specifically, and you just have this very simple, elegant loop as a solution. We just wonder, like, oh, did it actually change its behavior as we engineer it? And we actually solved a structure with Chris Garcia's lab, and we found out that it essentially was almost like a carbon copy of where the superantigen structure that we originally used. But what's really interesting here is actually the molecular interface of how that single loop is able to recognize the target. So on the lower right-hand side corner, you can see that the peptide, CLIP peptide, is an invariant chain. So all MHC2s that would have this peptide will be able to bind to this peptide. And then we just now have a atomic level picture of how the interaction is achieved. And it's actually really surprising, because now when you look at these structures, we realized that they actually forms very few contacts to the peptide. So initially, we were thinking that our sequence must be somehow docking perfectly direct to the antigen peptide. But then once we solve the structure, it turns out that it just has a few hydrogen bonds, and the rest of it is almost like a retractive interface, right? You scale back the side chain so you just don't run into it, and it's just making the reverse molding of that antigen shape. So initially we thought that, oh, this must be not very specific, right? How could this simple retractive type of interface be specific to the target? So we're imagining that they would be very cross-reactive to the mutations that's made to the peptide. And then we did this experiment, and it actually turned out to be very, very interesting. So the heat map here is showing the 15 positions, x-axis is the 15 positions on the peptide. And then the y-axis, the dimension is actually mutating every position to the 20 amino acids. And then the boxed ones are the native sequence. And it turns out that our binders is incredibly specific. So all the regions are in blue, meaning that the binding for those mutations are actually weaker than the native sequence, amino acid sequence. Right, so it turns out that even for this CLIP peptide, our binder is incredibly specific to that cognate target sequence. And actually, we have some data that I'm not showing here. It turns out that some of the targets, we can probably even recognize just very subtle differences in like almost angstrom level resolution. So with this specific CLIP peptide, for example, there's a proline at position 10. And that position, as I mentioned, this is a CLIP peptide. So all the MAC2 is polymorphic, but everybody would have to be compatible with this peptide. When you take the same peptide, put it onto a different MAC, actually our binder does not bind. And if you actually look at the peptide conformation in that new MAC that you put it into, it turns out that the only difference is just that one residue pivoting like by some angstrom level of difference. So it was actually really surprising how specific this system is. And I always thought that this is almost just like the analogy for a lock and key mechanism, right? But here, the development from our end is not the key. Like we actually don't need to unlock it, but it feels like the MAC molecule is actually the key jacket. And then the peptide, if you just think that the peptide is actually the key that fits into the MAC, because for the immune system to recognize these antigens, it's actually the peptide MAC pairs that has to be specific. And then what we're actually recognizing is that pairing of the MAC and the peptide. And what we're actually building, our tracer platform, is actually all the pins that actually can actually read out the specific pairing of the lock and the key. So this becomes quite interesting because when you have a system is reduced to that level of simplicity, right? So now, again, the picture in your head is that now we have this really complicated MAC, but they all look kind of the same, right? Every MAC, the mutations are actually, the polymorphism of the mutations are actually inside the pocket, so they load the peptide differently. But on the surface, they all look kind of the same. And we have this platform that engages it in the same binding mode every single time. And the only thing you have to adjust is just that one single loop. So when we reduce the problem down to this simplicity, it turns out that you could actually directly design what that loop would look like for any, well, maybe not any, we have one case. But at least now what we're trying here is to see, can we actually just take a random peptide that is, so this particular case is from SARS-CoV-2. We don't know what the MAC peptide complex looks like. But we literally just take the peptide sequence and then use everything we know about MAC and the peptide, and we just model it directly on the computer. Can we actually just design a loop sequence that will bind to that target? And it turns out that we can. So this is quite interesting because now it completely opens up this possibility of taking any, well, again, we have example one, but if this is extrapolated to different targets, we could actually just take any antigen, and then you can just build a binder for it directly on the computer. And nowadays, there are very advanced mass spec technology that is actually discovering these antigen peptides in high throughput fashion. And it's actually the hardest part to translate those information into diagnostic solutions is exactly the missing binder that all these targets would need. And we could potentially use this type of platform to achieve this type of goals. So I know I'm like running short on time, and maybe I'll take a few minutes just to very quickly cover some of the engineering modeling side of things. So for this MAC part, is essentially pioneered by my graduate student, Hao Tian, and then later on with Jing Jia to help develop this platform. So my lab actually is not super well known in the immunology field or anything. I'm actually better known in the computational protein design area. So we wrote this review article in Nature in 2016. But now we just have a new updated version of what the current de novo design protein looks like. This one is published in Nature Biotech this month. So you can kind of see that we now have this new capability to build these new novel proteins. And then I'm sure you've seen a lot in the recent literature how this technology has been used. So my lab specifically also developed a generative model where we actually try to use molecular design to capture flexibilities of proteins. So this is actually some of the unique, challenging area for protein design. And we actually want to apply this to antibodies. So see if we can actually, kind of like what I talk about it with the MAC story, can you actually just give me an antigen and we build you an antibody directly in silico so you don't actually have to inject into man animal or anything. So I'm just going to skip very fast through this area, this section. So essentially the idea is to mold these proteins to the target. And what we could do is to actually have this new generative model to capture the conformational space of sort of antibody or IG-looking folds, and then we can model those loops to sample around the surface. And then we were able to use similarly, this type of yeast display technology to screen for binders. And you can see that, at least with these random targets that we use, we can actually get binder for every single one of them. So, and this is a technology that's actually under the movie that Kathy showed earlier. And what we actually want to do, we're pushing for now is kind of like what I was saying. Current technology for building antibodies is all these tedious wet lab procedures. But we really want to do is to see if we can actually directly design it in silico just by doing this type of complementary surface modeling and using the cutting edge protein design tools. So with that, thank you for your attention. And I'll just, oh, sorry, this.
All right, so we'll move on to our next talk in this session, and it comes from Craig Levin, who's going to be telling us about the development of new technologies, actual imaging, and potentially diagnostic technologies. Craig is a professor in the Department of Radiology.
Thanks, Kathy. And as you can see, that picture up there was a few years back. So we're going to switch gears here and move from protein engineering to physics. And so that's one of the really nice things about molecular imaging. It's very exciting for me, and I hope my students as well, is this interdisciplinary nature of this program. And that's something that Sam really, really appreciated and nourished, and I thank him for that. So in particular, now it's late in the day, so if any of you are going to settle into your afternoon naps, there's just one point I want to make is that PET, positron emission tomography, can actually be used to interrogate multiple biomarkers in the same PET imaging session. At least we believe it can. In silico, it says it can, and so we just need to kind of build up capabilities to be able to do this in a real study. And that's what we're going to talk about. So we're shifting gears a bit now, more into physics. And if I can shift the, this thing is not shifting. I can give you the old next slide thing. Okay. And I wanted to start off, it's really hard for me to add to all the wonderful and beautiful comments people made about Sam and Aruna. Here's a picture of me and Sam when we were both much younger. I had just joined, I think, the program. I was actually me and Jung Hong Rao, who maybe is in the audience, so we were the first faculty that were recruited by Sam when he came to Stanford into the MIPS program, so it was really a great honor. And I remember the phone call from him and everything, so it was really great. But here you see us talking, and I guess what I really miss about Sam is actually sitting down talking with him. His voice was so unique, as you know, and just it was so fun talking to him. He was serious, but he'd make these very dry jokes, and just loved bouncing ideas around. So I really miss that about him. Here's the outline of the talk. I'll start with introduction to what we call multiplex PET or MPET. Excuse me, I'm sorry. So this actually slide here is from Andre. He took a prostate cancer patient and he did 6 different tracers. Some of them were interrogating different biomarkers associated with prostate cancer disease. He got, I think, the Ant Mini Award for this. I think it's an amazing study. Very hard to do this study, and unfortunately, they can't really do these studies regularly, where you do multiple tracers. In fact, it's even harder to do 2 tracers on different days, and so we'd like to, motivated by this, we'd like to be able to do at least 3. And we think we can do 3 at the same time. We don't think we can do 6, so we'd have to use some other method to do 6. So definition of multiplexing. In an imaging assay, it's an imaging assay that enables the visualization and quantification, interrogation, characterization of more than one biomarker in the same imaging study. Okay, so that's what we'll call MPET. And so the fundamental question is, can we transform this modality, positron emission tomography, or PET, from a single parameter imaging modality to a technology for multiplex biological imaging assays? And this is in order to get a more complete picture of the disease. Diseases are very complicated. You need to look at multiple biomarkers. Codex looks at many, many biomarkers. We can't look at that many, but maybe we can look at 3, and 3 important ones that are important for in vivo studies. Applications we have in mind, I'll talk about a few of these later, but here's some applications that we have in mind. And we're talking to various people about these different applications. Previous approaches to PET multiplexing, well, Andre Iaguru again has also looking at 2 different tracers of different biodistribution spatially and trying to eke out 2 different signals. Of course, there might be crosstalk depending on the type of disease. In this case, he was looking at FDG and sodium fluoride. I think sodium fluoride going to the bone, FDG going to the soft tissue, which had success. I think there were some papers published. You can also look at different decay times of the 2 radionuclides in a way, naturally you wait for one to decay and one to decay away, and then one to start decaying later, or you can inject them sequentially and things like that. Another thing you can look at is different kinetics, and Sarah Zhao in the audience, I think, maybe she's not here, but in our lab, she's looking at this where you introduce dynamic imaging data. You introduce a compartment model. You get parametric images from which you can get unmixed tracer images representing different biological features of the disease. In this case, an FDG image, an FMZ image, a different tracer. But there's often substantial crosstalk in these other methods, and so we wanted to come up with a clean method. It turns out that no method's perfect, and but we're hoping that it will be a little bit more clean. Now, SPECT, ironically, has been able to do multiplexing, although they don't really do it very often. Here's an example of an energy spectrum. Those peaks represent the energy of different gamma rays that are emitted in SPECT. And in SPECT, you naturally can distinguish different tracers and therefore different biomarkers in principle by looking at different energies of the emissions. And but clinically, at least until recently, SPECT has been had much lower sensitivity and spatial resolution for PET, and so it's not as attractive, although it still is attractive in some applications. So the need for PET multiplexing, or what needs for PET multiplexing. Here you see a patient. This is my art there, so you can see the patient's head and their shoulders and little nose there. Feet are going into the page. So I explained this because I showed this to my mother and she didn't know what that was. And so, and then we see these squiggly lines coming out. pairs of photons, red and green, showing tracer 1 and tracer 2, indistinguishable in a normal PET scan if you have two tracers because you're looking at the annihilation photons coming off. They all have the same energy, 511 keV. They come off oppositely directed, but they are at the same energy. So you can't tell the difference between tracer 1 and tracer 2. And this is why conventional PET can only do one tracer at a time. So that's out. You can't do that. But what you can do, you can, if you have tracer 1 being a pure positron emitter like we just saw, but tracer 2 is a positron emitter that also emits a gamma ray simultaneously. They call it in cascade, so that wherever that positron is emitted, we also have a prompt gamma ray. So then that's going to result in a triple coincidence, 3 photons emitted, 2 of them being the 511 keV photons oppositely directed, and then 1 of them being that prompt gamma ray that would then produce a triple hit in the system and therefore be able to distinguish between a 3 and 2 photon, one tracer and another tracer. And I'm going to be showing you the radionuclides mainly in this talk, but you can imagine the radionuclides being labeled, labeling a ligand, so it's a radioligand that's targeting certain receptors, so you can report receptor status, and you can do that in principle with 2 tracers, maybe up to 3, we think. Anyway, so we call those positron gamma tracers or prompt gamma tracers. So we published the first paper studying this method. Actually, it was an experiment that we did in 2011, a long time ago, demonstrated that this could work. You could separate 2 different tracers this way. And we got 2 patents out of it. One of them, look at that second patent. The second author on there is Sam. So Sam was very interested in this idea, and this is one of the reasons why I'm presenting this. Not to mention, we think this is a killer application for the new cyclotron to develop new radionuclides that are prompt gamma emitters. And we'll see not every radionuclide is a prompt gamma emitter. Here you see one example of a positron plus prompt gamma emitter, scandium-44, we think we can use in diagnostics and theranostics. In fact, working with Corinne Benau, who we heard from earlier, on a project I'll talk about a little bit later. Cobalt-55 is also very attractive. It has a longer life, so for immune studies where you want to look at immune response, you need a longer half-life. Cobalt-55 is very attractive, so it's a triple emitter. I-124 is another one that's attractive, although you see in the right the table of, I don't think these, is this table showing the different prompt gamma emitters, emissions. You get 4 of them, and really the most common one is 603, quite close to 511, so it's not ideal if it scatters a little bit, and the patient will show up in the 511 keV window, so it can be tricky to use that. Also, as a counterexample, zirconium-89, we love zirconium-89 for immunopet. Zirconium-89 is not a positron plus prompt gamma emitter. It does emit a gamma ray, but it's not prompt, so you won't get a triple coincidence. So alas, we can't use zirconium-89, but ironically, we're using zirconium-89 as the pure positron emitter in the study, and then another positron plus prompt gamma emitter in the same study. Okay, so needs for this, to be able to do this. We need to have 1 or more positron-emitting radionuclides that emit a gamma ray promptly. Okay, so you get triple coincidence. You need a PET system that it will accept triple photon coincidences. Conventional PET systems don't. Some of the newer systems do accept triple coincidences. So we need to be able to see triple coincidences, which are normally thrown away in the data sets of a conventional PET scan. We need a PET system that will accept up to 2 MeV of energy photons, because some of these attractive radionuclides that are positron plus prompt emitters emit up to a close to 2 MeV gamma ray. So that's really hard for some of these PET systems to be able to collect energies up that high, but it can be done with proper design. And then a PET system with high detection efficiency for 3 photon coincidences. We know that we collect a relatively few number of these 2 photon coincidences. Ordinary PET has a very low sensitivity, so triple coincidences, a factor of 10 times lower sensitivity, so, or even more. So you have to have a very efficient system. So geometries, speaking of which, and some results. So here's again, my art again. You can tell it's a person, probably a man, a little cartoon drawn in PowerPoint. going through a PET CT machine. In green are the annihilation photons that you want, the true ones that you want to collect. Then sometimes one of the photons in the pair doesn't get detected because the PET system doesn't cover all the way around the patient. And then you also get cases where neither the annihilation photons are detected. So if you imagine, that's 2 photon coincidences. Imagine 3 photon coincidences. You'll almost get no coincidences in a normal PET scan. You will if you wait around, but compared to the 2 photon coincidences will be much, much lower. So attractive geometries, we've built a brain-dedicated geometry. The nice thing about that, you bring the detectors in closer, so you have better geometric efficiency, better chances of photons being emitted to traverse detector material. And here you see our first generation, what we call PET coil, that goes inside of an MRI machine. That's what we call the PET coil, because you put coils inside of an MRI machine, even though PET system is not a coil per se. And here you see our second generation, much more sensitive system that some of you that toured the lab yesterday got to see this system and it actually working. Oops, and now I did switch so many here. Okay, I think that was it. Yeah. So let's see if I can keep it. Oh, it wants to jump. I can go backward. Okay, so there it is. It comprises a ring of 16 detector modules, and there you see another picture of it collecting information, in particular energy information and timing information. And this energy information, timing information, if precise, will help us to properly classify 2 and 3 photon events in a multiplex PET study. You can imagine all the different types of random photons that occur where you can misclassify a triple coincidence as a double coincidence and vice versa. The energy and timing help to resolve this problem. What we want to do on this system is actually add an end cap to the system. It's hard to see in that picture, but it would actually fit right in the back. Putting an end cap here, that's going to improve the geometric efficiency for collecting 3 photon coincidences. And West Foster in the back, you see occasionally laughing and over there. You can see that picture. You can look at this picture and see him there. So put him on the spot. Anyway, he did this some simulation study, showing what the efficiency we can expect with and without an end cap. And it turns out that if you build an end cap, you should also make it position sensitive, not just a slab detector, but it's also part of the imaging detector. This here. And that will improve your 3 photon coincidence sensitivity for various radionuclides that emit those 3 photons. Just some more studies of how thick you need to make that detector. In a standard PET system, using something like 2 centimeter thick crystal in a PET study, the high energy photons will go right through it, and maybe they'll scatter once in a while. So we may be making a thicker detector system for the top end cap of that brain imager. And Sarah Zhao again did some simulations. So this is in silico of what we call a heterogeneity phantom. So it's spatially and in principle biomarker heterogeneity phantom where we have, we have one lesion has both signals, in this case I-124 and F-18 labeled radioligands, or ligands I should say. And then here we have F-18 only and I-124, so spatially heterogeneous. And then we have an overlapping region here. You can see the different results when we just do the raw triple coincidences. Raw triple coincidences. You can imagine that only the I-124 signal would show up, but we can improve on that with some line of response identification algorithms that Sarah has developed. We can better hone in and estimate the I-124 signal, which is on the left here, or on the right here, and then the F-18 signal on the right. So this is simulation, looks very promising. The accuracy is decent. We still have to improve this quite a bit, and then we, more importantly, have to build the system and actually get real measurements to prove it out. Another really super attractive system geometry for us, and maybe is a killer application for this total body system for multiplex imaging, is this total body system, long axial field of view system that, like the brain imager, it's covering what you want trying to image, the geometry, all the possible directions the photons can be emitted. So this long body geometry is very attractive for this MPET application. So in particular, 128 centimeter field of view is really a sweet spot for a very high efficiency, or relatively high efficiency But again, we need to be able to accept triple coincidences, which the system I think will be able to do, and then accept up to 2 MeV energies. A little questionable right now, possible in a future system if we pay GE enough. Example applications. I just want to quickly go through applications. We are funded by the Stanford BioX, an internal funding mechanism, as well as the Falk Medical Research Trust Foundation. Both those projects have Dr. Jong-Hong Rao and Dr. Ronald Levy on this, looking at different biomarkers for cancer immunotherapy. T-cell activation, infiltration, cytotoxicity, exhaustion. In particular, infiltration and cytotoxicity is our focus right now, and the main hypothesis is that if this T-cells and their cytotoxic activity can be imaged simultaneously, that's a more important information for determining a response. And we're going to be developing tracers for these two biomarkers. Another project funded by the SNMI Mars Shot is in the realm of breast cancer. We know that breast cancer also is a complicated disease, requires characterization of multiple biomarkers. In this case, we hope to do it simultaneously. We'll start off with looking at estradiol as a ligand for estrogen receptor positive breast cancer. So breast cancer that overexpresses the estrogen receptor, and that's a good ligand for that. The standard ligand for estradiol is labeled with a F18, so we're going to knock off that F18 or label it with a triple emitter, scandium-44. So challenges. This is not a slam dunk. We have to properly classify those 2 and 3 photon coincidences, especially if we're trying to do up to 3 tracers simultaneously, to be able to unmix them cleanly. Labeling new and existing tracer molecules and expecting them to have the same biodistribution as a conventional label like F18, that's really a sort of a Mars shot. Often low probability of gamma ray emission. So sometimes you don't always get a prompt gamma ray, so we want to pick those radionuclides that emit a prompt gamma ray very often as the positrons emitted. And then sometimes you get more than one prompt gamma ray emitted, which I showed you, iodine-124 as an example. Some of the gamma rays are high energy and they don't stop in the material that you're using in your detector system. So that's something we have to deal with, figure out how to properly get those events. Perhaps the ones that come in obliquely, they're seeing more crystal, for example, will have a better chance of seeing a photopeak. Often large positron range of these non-standard positron emitters, giving us a blur in spatial resolution. Okay, so we have to deal with that as well. And dose, we have to maintain the same dose. We can't just triple dose a patient, so we have to be able to deal with the lower signal noise ratio. Fortunately, because of AI, there's methods to do that. So I want to say quickly, to give you an example, so labeling new and existing tracer molecules. Now, Corinne is working in her lab and trying to, she's been very kind to support this project, and she's working on labeling estradiol with scandium-44. So nobody's ever done that. It's always been labeled with F18. Somebody did label with technetium-99, so we looked at that paper. They had some successes. Here's this estradiol molecule. Oh, sorry, so now we're switching gears. A little bit of chemistry, if I can do a little chemistry here. So here's estradiol, a molecule. Again, it's going to be targeting estrogen positive receptors, or estrogen receptor positive breast cancer. F18 estradiol, they label it right in this little spot here. They put an F18 there. Works great. It's been evaluated in patients. But now what we can't put a scandium-44 here. It's a metal. We have to chelate that metal, and we can't put a chelation here. We have to put it down here. And also, for those of you that are chemists, when you do chelation, scandium-44, it's huge. So is that going to have the same properties as the F18 labeled estradiol? Well, we've given that job to Corinne. So, yeah, thank you. So Corinne's working on that. But if anybody can do it, she can do it. So yeah, right? Yeah. So she's smiling here, but after she works on this project. But anyway, no, she's got some very talented trainees that are working on this project, and we're just so thankful that she's even trying this, and I think it's a possibility. But what we have to just show is there's going to be, I don't know if it's going to have the same exact binding affinity as the F-18 labeled molecule, but if it does show enhancement, that's kind of what we want. So we're hoping for that, and so will it perform the same? We don't know that yet, but as long as it shows some preferential uptake, then that's what we want. So in summary, it's still a moonshot. Maybe it's not a Mars shot, but it's a moonshot to get this going. Lots of things have to come in place, but if we could do this, we'll be able to unmix we'll be able to unmix signals from multiple biomarkers in the same PET imaging session. There's lots of system design needs that are necessary. Chemistry is got to be studied. Simulation results so far look very promising. We still have to get the system built and show it working in animals first, probably, and then humans. And then we have a number of important applications really motivating this work. The physics is all cool, but the real motivation is to characterize disease, get a more complete picture of disease by looking at multiple biomarkers in the same study. Being able to do this in sequential studies is just not possible in the clinic. So thank you very much for your attention. I just want to, before we go, emphasize that partners here, radiochemists, biologists, animal models, these are all very important, and also physician scientists to guide our appropriate applications.
If you deposit all the energy.
Thanks very much. Yeah.
Fantastic presentation, Craig. This kind of goes back into the research. When I was a grad student in the early 2000s, the group I was in, we put out a paper called Addressing the Third Gamma Problem in PET, where we were worried about, do you get image degradation from the extra gamma from isotopes such as I-124? So we were worried about it.
Yeah, it's called dirty emitter.
Yeah,
dirty emitter, right?
Dirty
Yeah.
positron emitter, yeah. So my question is...
We're turning dirt into water or whatever. Yeah.
You are.
Yeah. I didn't say wine though. I said, okay, yeah. Gold. Okay, yeah.
So my question is, that was in the days of 2D PET, where we didn't have all these oblique angles. Scatter wasn't as big a deal. And I just wonder, when you start multiplexing, have you modeled, I think it's going to be less than half. You're going to have to go even lower on your administered radiopharmaceuticals, because you're going to have that scatter component, you're going to have that extra gamma, and your count rates are just going to be through the roof. So have you looked at like the sensitivity you're going to have in vivo as a result of this multiplexing?
Yeah, so no, we definitely want to have to experiment with the dose. The majority of the dose, as I understand in a PET scan, is actually the positron annihilating, because the photons, although they scatter in the body, a lot of them get out. And so majority of the dose, but we do have to model things like the gamma, the prompt gamma interaction in the body. But ideally we're using something that has been a radio label for another tracer and studied in humans, and so it won't be the first time that the radionuclide has gotten into humans, just a different application.
Yeah, my concern isn't the dose. My concern is the image quality, because you're worrying about count rate saturation.
Yeah. I mean, so if I understand correctly, if we say we're going to inject 3 tracers, but we reduce the dose of each by a factor of 3, then we have a noisy image. Is that what you're?
Your image is going to be noisier because you have those prompt
Yeah, and then
in
now
there as
there's
well that are contributing dead time to all your detectors.
Yeah, and that's a very good point, and we haven't shown it, proven it yet, but there's a lot of work out there, and we are looking at in other contexts where you can actually reduce the dose that you give and actually get the same image quality using AI methods. And that's kind of what and so there's a few companies have been established, including one that came out of Stanford. So we'll interact with them, we'll look at literature, we'll do our own work. And so I think that actually I'm least worried about making a good image out of 1/3 the dose than I am all those other things. Yep, but it's a good point.
One more question.
Yes, Fresgi. Fresgi was one of the reasons why I'm here is because of Fresgi. Fresgi was my first postdoctoral fellow. So, family.
Thank you.
Yeah. Okay, now because I gave you that plug, make sure it's a nice question now. Friendly question, Ken.
Was a triple coincidence. How do you deal with the resolution, the line response? Which
Yeah,
one
now,
do you
good question. So remember the positioning of the event is gotten by the annihilation photons. That triple coincidence helps us characterize it. So we're not actually trying to position that third photon. We don't know actually, we don't need to know where it actually interacts. We just need to know it came from the same pair as that other annihilation photon. All the imaging is done the way we normally do with annihilation photons and line projection operations. Good question, though. Oh, one more question there. Yes.
I have a question regarding the scanner you mentioned. Like you said, you were going to get a long axial field of view scanner from GE. I think.
We're praying. I mean, no, it's not quite at level prayer. It's above
Okay.
the one step above prayer. Okay. Okay.
Like this, the picture you showed was 128 centimeters field of view. I think.
Yeah.
I think that's applicable to the GE system.
Yeah. Yeah. Oops.
Do you have any data? Does it work with the prompt gamma? Actually, do the time of flight resolution, which is like AI-driven, enough to characterize this prompt gamma?
So you're asking me if the capability of that system that we hopefully will get will have that capability?
Yes.
That's a perfect question for the next speaker. Actually, it's amazing. Did you guys arrange that ahead of time? Anyway, that's perfect. Yeah, maybe Flores, should I answer that or where's Flores? Yeah, should I answer that or will you answer that in the next talk? Anyway, we hope so. Yeah. We hope so. And if not, we'll do our best with that data to try to squeeze out what we can. And then we'll also, if the data looks good, we'll convince this gentleman to ask his higher ups to increase the money in that program, and then they'll do a perfect design for us. But we have to convince him first, and he's not that easy to convince. So.
Perfect. Perhaps I can comment on the dose reduction question. I mean, if you use this long axial field of view system, I think you will be very easily go beneath 1/3 of the dose. I think that's very easy to do.
Thank you so much. All right, we're going to move on to our last official talk in the session, and this is going to be followed by closing remarks and then by a bus that's going to take us to the facility. So we're welcoming Flores Jansen, who's the Chief Scientist in Molecular Imaging at GE Healthcare, and he's going to be telling us more about next generation technologies.
It's great to be here. It's an enormously talented group of speakers that I'm following, so I'm feeling a little bit of trepidation here. But I hope to show you a few things that are new, a few things that are familiar, and maybe answer the outstanding question. So this chart here was actually shown by Andre earlier. The transformation that has happened in molecular imaging over the last 20-some years has been astonishing. I remember meeting Sam back in 2003, 2004 when he had just come here, and he had this incredibly sharp vision of the future, and I think he would be very proud to see how far along we have come, even though we are all aware that there's still a long way to go. And I'm happy to be one of the people providing shovels for you to do the gold digging. So on the left here you see a ton of new tracers that are in various stages of commercial or pre-commercial development. In the middle, those tracers, those therapies are going to bring a lot of economic activity into the world of nuclear medicine. But at the same time, because these are all very much precision medicine focused tracers, we are going to have a significant uptick in the demand for imaging. And so with that in mind, we see a changing procedure mix, we see a rapid growth, we see additional indications, but at the same time we see limited or finite healthcare resources, not only the money that is available, but the people that are available, the time that is available. And so with those constraints in mind, we started asking ourselves, what is a PET-CT system going to look like that really plays into, that leans into both those opportunities and those challenges? And before I tell you more about what that system ended up looking like, I want to give you a little bit of an insight into my understanding of how to build PET systems. Very frequently, you will see people who have a new PET system focus on a specific thing that they did particularly well. And if you just try to word cloud it, there are a lot of buzzwords, a lot of opportunities, and each of these things can help make a scanner better. But in this, I use this analogy of triathlon. If you know that somebody can run a very fast 100 meters, you kind of know that's a good athlete, but you have no idea if that person is going to win the Ironman, because they may not be able to balance on a bicycle, they may not be able to swim, they may not be able to keep that sprint speed going for an entire marathon. So the only way that you can really tell how good a person is at the Ironman is to look at the podium. Where did they end up at the end of the day? And so when I think about imaging systems, I think about all the different ways that things interact, and the only real way to evaluate, to see where you ended up, is to look at the images, which we will do a little bit later. But let's start by just asking the question of image quality optimization, where I think of a good image as an image that has low noise, good contrast recovery, it is quantitatively accurate, low artifacts. And there are a number of knobs that you can turn, and Craig and I can talk at length about this, but we don't have the time, and nor is this an audience of physicists, and I apologize that you're getting 2 physics talks in a row here. But you know, system sensitivity, how good is it at collecting the radiation from the patient? The timing resolution, how well along the line of response can you position an event? The spatial resolution, how well do you know where the event ended up? The algorithms that you use for reconstruction, for correction, for image processing, all those things matter enormously. And so I'm just going to look at a couple of the trade-offs here, and in particular, I want to look at the trade-off of sensitivity and timing resolution. Because a long time ago, Tomitomo and Brombudiger postulated that having better timing resolution is like having greater sensitivity, and up to a point that is true, but it is very interlinked with actual count densities, it's interlinked with algorithms you use and everything else. And of course, one way to improve your timing resolution is, for example, to make your crystals thinner. When you do that, your spatial resolution gets better, your timing resolution gets better, your sensitivity goes down, and you can ask yourself the question, did you win or did you lose when you did that? Because real engineering is always about trade-offs. So we did a simulation, and we simulated as accurately as we could 3 systems that had the same basic geometry, same ring diameter, same crystal size and everything else, but we gave them different effective stopping power and different timing resolutions. So I'll call them systems A, B, and C, and system A had the lowest sensitivity but the best timing resolution. System B had double the sensitivity but a timing resolution that was almost exactly twice as big. I can never know whether you say it's bigger or smaller, better or worse, because these things are inverse. And the third system had triple the sensitivity, but was a non-time of flight system. And then you can simulate a NEMA IQ-like phantom, but because the large spheres in a NEMA IQ phantom aren't very interesting, we decided to simulate 6 small phantoms that gave us some oversampling, better statistics, etc. And if you just do a conventional OSCM reconstruction with points per function modeling, these are the 3 images that you get. So the lower sensitivity system with the good timing resolution gives you a somewhat noisy image, and the contrast to noise ratio is about 7.1. If I double my sensitivity, but I also make the timing resolution a factor 2 bigger, I actually get a very similar contrast to recovery and a somewhat better contrast to noise ratio because my noise has gone down. And if I take the third system and I use the same, roughly the number of iterations that get me to a similar contrast recovery, my contrast to noise ratio is a little bit worse. But that is assuming all other things are the same, and they are not. So the next step is I'm taking just these two systems here, and I'm adding Q-clear reconstruction. It's a regularized reconstruction that allows you to achieve greater convergence while controlling the amount of noise. And you can see both these systems improve in their contrast to noise ratio when you do that, but this system actually benefits more from it because I can now do more iterations. The problem with non-time-of-flight reconstruction is that things converge more slowly, so you want to do more iterations, but then you get noise. With Q-clear, I can do more iterations without increasing the noise, and I get essentially a better contrast to noise ratio than even the pretty good time-of-flight system that has somewhat lower sensitivity, and a lot better than the signal out here. I'm not done yet. I will come back to this chart later, but this is just showing you that one characteristic doesn't necessarily tell you what the system is going to do because you have to look at the totality. So in this case, you see improving sensitivity and sacrificing timing resolution actually made things better. Recon algorithms also can be a big differentiator. So having spoken about sensitivity as a parameter that can clearly be helpful, let's now tie that to axial field of view, and this will get back to what Craig was showing earlier. So if I take a normal-sized patient and I take a 2010 era PET scanner, which is about a 15, 16 centimeter axial field of view, I need to scan multiple positions, or continuously if you want it, in order to cover the isosceles region. So if I do 4 minutes per bed and I do 8 beds, that is about 32 minutes of scan time. But if I look at the physics of photon detection, if I make my scanner axially twice as big, I basically have roughly double the solid angle. So because the photons come out in a random direction, if the solid angle in which I will catch things is doubled, then I will get roughly double the sensitivity in that slice. But also my field of view gets bigger. So the total probability of radiation from somewhere in the patient being detected goes up by a factor 2 because of the solid angle, and another factor of 2 for the length. So it gets quadratically bigger. That quadratic relationship kind of breaks down when you get to very big scanners. It works very well down here, then it slowly goes more linear. So if I have a bigger scanner, now I could perhaps cover the same size of patient in half the number of steps. And because my sensitivity per slide doubled, I can cut the time per bed position in half. So instead of 8 times 4 minutes, I now have 4 times 2 minutes, and the whole scan will take maybe 8 minutes for similar image quality. Make my scanner twice as big again. And now I have 2 bed positions of 1 minute each, and I'm down to just 2 minutes. That's a big deal. Scanning faster can be enormously helpful. If you say, I have a need for greater throughput in my department because I have that demand of scans coming up, having the ability to scan more patients is very useful. If I can scan for less time, my patient is likely to move less. If I have a patient who, for whatever physiological reasons, is having difficulty lying still for a long time, being able to scan them in 2 minutes may be the difference between having a scan and having no scan. So Just being able to scan the same thing faster is important, but I could also, and maybe as well, lower my injected dose. Because if I have so much more stopping power, so much more sensitivity, I can get the same image quality in a shorter time and with less dose, which not only benefits the patient, this not only benefits maybe my bottom line because I have to buy less tracer, but it also benefits the operator who stands next to these patients all day long. And if I'm doubling the number of patients that they stand next to, but I have the injection in each of the patients, and then I didn't affect the dose to the operators. And of course, image quality is going to be positively affected because image quality is very tightly connected with count density. No matter how good the resolution of my scanner is, if I don't have enough counts to support that image, then the resolution doesn't matter. So I need enough counts, and in fact, sensitivity is the key to achieving resolution, because without counts, you don't get resolution. So imagine you go even bigger, 128 centimeters, there's that number again. If you scan 128, if you make your scanner 128 centimeters, you won't actually get 128 centimeters worth of usable field of view. There's a little graph here showing you the approximate sensitivity per slice. This is a geometric simulation, so it's not super precise. You see, as you increase the field of view, 32, 64, 96, 128, sensitivity per slice increases. Now, in this case, I am showing the sensitivity for 1 meter of scan range, which is going to be sufficient for most situations. And as you can see, if your field of view is 96 centimeters, then you have nothing at the end. If your field of view is 128, you still have a very decent number all the way out to a meter. I'm not saying the field of view of a 128 centimeter system would be a meter. It's going to be a little bit less than the total. This graph would basically go to zero. So how quickly you lose quality at the end depends on how close to the edge you're trying to go. Okay, what does that have to do with GE? Well, we developed a new scanner called the Omni Legend. You saw a picture of the actual scanner on the first slide. And inside that scanner is a new detector. It's a scalable detector. You see it's kind of outlined here. It's actually built with BGO scintillators for reasons I'll explain in a minute, coupled to a silicon photomultiplier array. And it is very, very scalable. Why did we use BGO? BGO has unrivaled stopping power. It uses bismuth with a very high atomic number of 83. That gives me a ton of stopping power. So if I have 20 millimeters of LYSO and I switch it for BGO, I immediately get a 28% increase in coincidence stopping power. But if I make my crystal a little thicker, I get a total of a 75% increase in coincidence stopping power. So thick BGO stops a lot more radiation. Again, if you do the kinds of things that Craig is wanting to do, where he's going to try to stop high energy photons, it becomes even more important that I have high Z scintillators. Secondly, this BGO detector has very good energy resolution. People typically associate BGO with energy resolutions in the 16 to 25%. We are measuring around 9.8%. How does that happen? That silicon photomultiplier array very efficiently collects the light, much more efficiently than conventional photomultipliers. Result of that is really great energy resolution. So sorry to say, in the standard configuration, Craig, we have narrowed our energy window so much that that 602 keV peak from the iodine is actually out of the window. That's great for conventional imaging, not so good for triples, but we can talk about what we can do. Thirdly, because I now have this very high density decoding of the crystals, I can make my crystals smaller, and I can get actually really good spatial resolution out of this detector. This is a Dorenzo Phantom. That's a 4.8 millimeter rods. They're beautiful and clear. Finally, having made this detector out of smaller blocks than previous generations BGO detectors, I can now get very good count rate performance. The noise equivalent count rate curve is one way of showing that. The peak noise equivalent count rate is now 500 kilocounts per second. That is higher than our highest LYSO-based scanner, which hits 450. That's the 30 centimeter field of view DMI6 ring, which I believe you will be getting. And it's much higher than really any other scanners on the market in what I call the 30 centimeter class. So plenty of sensitivity at the low end, plenty of counting capabilities at the high end. Now, scalability is really outstanding on this system. This is one building block of the detector. And as you can see, I have individual units that go in there. There are 6 slots. So in principle, I can slot any number. Commercially, right now, the 3 and the 6 are being shipped. Other configurations may be imminent. But you can see here that building block can actually be stacked. So you see one of them here. It is designed for a second one to fit right alongside it, giving me a 64 centimeter. And it is designed to allow me to take 2 of those and make a 128 centimeter system. That is not currently a product, and FDA and other people will tell me it may never be a product. But there are such machines in labs today. And I'm looking forward to the day that it'll be here. Okay, what does that field of view potentially do for you? At 46 kilocounts per megabecquerel, the Omni Legend 32 has the highest sensitivity by a long way of any scanner in, again, what I call the 30 centimeter class. The DMI 30 centimeter system is the closest you get at 30. Other systems are, struggling to even hit 20. When you double the field of view, you almost double the sensitivity. When you go to 128, you get another big boost. I don't want to put numbers on there again because as this is not a cleared product, I don't want to get into that story. But that curve goes up a lot, and consequently, those time trade-offs go down a lot. Great, but you don't have time of flight. And we know from the earlier slides, time of flight is a helpful thing to have. Not having time of flight is missing a piece of information. But we traded off missing that piece of information in return for having a lot of sensitivity and all these other good things. Both in our MR and our CT business, we have had opportunities to explore how we could get good images if we were missing some information. MR wants to go fast, skip points in K-space. What we call Air Recon DL gives us good image quality. In CT, you want to lower the dose, you're missing information. True Fidelity gives you great image quality. So we said, can you do the same thing? The answer is yes, you can. You can train an algorithm using both non-time of flight data and time of flight data to say what was missing in this initial data, but that should look like that. How do I create that transfer function? So having trained that algorithm, we can take an image that looks like this, where you have this barely visible lesion, and turn it into an image like that, which is essentially indistinguishable from the time of flight. When we do blind reads, people either can't tell them apart or they prefer the Precision DL image. It comes in 3 different strengths. So here you have a non-time of flight reconstruction, and then I add either the low, medium, and high. And as you look at this cluster of tumors, you can see how it becomes clearer. If you look in these axial views, you see the same thing. You see some tumors that are barely perceptible there becoming very clearly visible. As you go left to right, you get slightly more noise. That's kind of normal when you try to push contrast. But if you look at the quantitation now, it turns out that if you compare non-time of flight to time of flight, you typically get a reduction in SUV max. And as you go through the different stages of Precision DL, you can see that the bias goes effectively to 0. Okay, so now let's take Precision DL and apply it to that chart I had earlier. So I've added an extra row here, because as it turns out, System C looks a lot like the Omni Legend. This is 45 counts per second, 46, okay. And now if you look at this, you get a contrast to noise ratio going all the way up to 9.7. So you get a significant reduction in noise, increase in contrast using this system. What has happened then is the Precision DL has essentially bridged the gap between the non-time of flight and the time of flight recon, but I got a lot more sensitivity. So I gave something up and managed to use artificial intelligence to get it back, but the sensitivity boost that I got in return remained. Okay, now let's look at some images. And the images I'm about to show you were all from University of Cardiff, Professor Bradley, who pushed his scanner to make what I believe are some of the best images. He does not have a very high patient volume, so he scans slightly higher doses, slightly longer times. And I think that that gives us a glimpse at what a larger field of view scanner is going to give us. So here's the first example. This is a patient with a, this is a medium-sized patient. The scan was 16 minutes and just beautiful, beautiful image quality. If you now take that same data and you virtually reduce the scan time by taking 75%, 50%, or 25% of the total events in the list file, you can see that while the image quality does degrade a little bit, even that 4-minute scan is really quite good. And I have a couple more examples. Here's a much larger patient. As you know, time of flight technology typically is more beneficial if patients are large. This patient is very large. She is 129 kilos, but only 1 meter 58 tall. So she's like a, physicists like to talk about the spherical cow, right? This subject is nearly spherical. But again, here's a 16-minute scan, taking it down to 12, taking it down to 8. You maintain good resolution, you maintain reasonably low noise. There's another patient, a BMI of 36, and again, 20 minutes, 15 minutes, 10 minutes, 5 minutes. You reduce the scan time by a lot, but the image quality remains pretty good. Okay, so as I said, I think that gives me an idea that if I get that scanner with much higher sensitivity still, I can move all those times down and maintain that great image quality that I have with the longer scanner. But I just want to get back to motion correction for a second, because it came up in an earlier talk, and this is, I think, very relevant. Patients breathe, which is a good thing, but when they breathe, their respiratory motion can blur my image. We used to have a hardware solution to correct for that, and that hardware solution is really, people ended up not using it. So we went to a fully transparent method. We call it motion-free. It analyzes the PET data on the fly. It finds the respiratory triggers, and it corrects for them. And so you can go from this image to that image at the push of a button. And this was an 8-minute scan in 2 beds. Essentially, half the counts were thrown away to make a decent image. But because you start with such high sensitivity, you really don't get a big noise penalty for doing that. And as you can see, there were some small lesions that were virtually invisible with the respiratory motion present that are recovered when the respiratory motion is absent. Now, people sometimes say, why do you throw away those events? Why don't you just do it properly by taking all the counts? It turns out that is actually quite possible on our system. You see here a little movie if I, oh, sorry, that should play. Thank you. So you can see here that you can do gating and you can get that movie, and in fact you can use optical flow methods to turn it into a single image. But that ignores this phenomenon, and that's actually the same patient that we showed a moment ago. This is the actual respiratory trace that I recover from the data. So I can see that at end expiration, this patient gets to the same place. But at end inspiration, the amplitude is all over the place. And if I look at this movie here, you can see this tumor go from very blurry to very crisp. This is all offline analysis. This, as such, is not available on our system. But it shows you, first of all, that it is possible to get a respiratory trace similar to what was being shown for the radiation therapy at some point in the future. That might be a part of our product. But also that a respiratory gating may not be the best way to collect this data if you didn't just take the quiescent phase. That is my time right there. 0. Okay, thank you for your time. Do we have time for questions? Yes.
Precision DL, low, medium, high. Can you say what you're doing in the training that's different, or?
Yeah, so I did not mention it, but we train with different target images. So, you know, some people scan for longer and they try to get really sharp images. Some people scan for shorter and they crank up their beta for their reconstruction a little bit. So you can train to target images with different betas. And you know, starting with images that have different count statistics, etc. And so that is what we did. And I like to compare it to an Indian restaurant. You know, if you go, you say, you know, do you like mild, medium, or spicy? And so if you use precision high, it's like saying you like your food spicy, and you better be ready for those 3 chilies because it's going to hit you.
Hey, Flores. Thank you so much. This was a great presentation, a lot of exciting stuff, but I want to challenge you a little bit. So the first one, 2 questions. First one is, you know, you showed your plot of expected growth in nuclear medicine. Most of the growth, I see a couple of big growth areas, the ones that are going to grow the most, but basically PET-CT might not be the better solution. I'm talking about neuro and cardiac.
Yeah.
It's very difficult to basically look at these 2 target applications that you're going to see there, or the many target applications you're going to see there without having basically an MRI component. So that's the first one question, why stopping there? I think I know the answer, but I'll let you elaborate. And then the second one, I see that you're trying for the VGO crystals, trying to basically rely a lot on artificial intelligence type algorithms for basically compensate for what you're missing. But I see a lot of your results are basically based on FDG data. And one of the things about FDG being not that specific is that there are a lot of features in the images that the DL algorithm can use. The minute that you start
I'm having a hard time understanding you. Could you slow down a little bit? I'm sorry, it's the acoustics.
First part, you got it, right? Why
Yes.
why not MRI? Okay, second part, relying very heavily on the artificial intelligence. The algorithms that you're using, I see a lot of FDG data, which being very non-specific, you have a lot of features that the algorithm can basically borrow from for learning. For some of these more novel tracers, they tend to be more highly specific to some of the targets.
Yep.
So
Okay,
how
got
do
it.
you plan to basically go around that? Because probably
Okay.
the DL stuff is not going to work.
Right. So first question, some of the growth is in neuro and cardio, and PET-CT may not be the right thing. True, but there's plenty of growth left in oncology, even if these other things did not migrate to PET-CT. But I do believe PET-CT and to some extent PET-MR will pick up some significant things. In fact, you know, Matthew sitting back there has done a tremendous job making brain imaging on our PET scanners much, much better. I didn't have time to talk about that today, but it's a thing that you should come and see sometime. Second question, today Precision DL is cleared by the FDA for FDG oncology only. We do have a lot of data for other tracers and are in the last stages of development of a multi-tracer capable model that will greatly expand the capabilities. But again, because that is not currently a product and because I had a finite amount of time, I did not want to get into those details. But we are aware and we are developing a second generation algorithm that will specifically have certain other tracers in its indication. And I believe after that there will be a third that will be even more general and that will be able to handle tracers that it has not seen before. But that is technology in development that may or never become product. It's not been cleared by the FDA for all that stuff. Legal disclaimer. All right.
Thank you so much.
Thank you so much for your time.